Hello, good morning. Hello, everyone. Uh, as you can guess, I'm not John Zehr, but I'm going to introduce you, the speaker for today. It is really a pleasure for me to introduce Professor Jonathan Zehr from the University of California, Santa Cruz. Professor Zehr, as you might know, he's a microbial ecologist that has dedicated his career to try to understand the role of microbes in the nitrogen cycle across different aquatic ecosystems, including freshwater, hypersaline systems, and also in the ocean. So his primary focus is on marine nitrogen fixation in the oligotrophic ocean, and I personally can't think of anyone that had contributed more to our current understanding of marine nitrogen fixation than Professor John Zehr. He has led quite a few groundbreaking discoveries over his career, including the recent discovery of probably the first known, I'm not spoiling the talk, the first known nitrogen-fixing organelle in an eukaryote. And I must say that I had the fortune of working with John for a few years there in Santa Cruz for 3 years. And I'm very thankful for that because that was a really, really good time, and it was really a source of inspiration for my science as well. So I'm gonna keep my introduction, stop my introduction here, and so please join me welcoming Professor Zehr to the stage.
Well, thank you, Fran, for that very nice introduction. It's nice to be in Barcelona again. I was here right before COVID as a guest of Sylvia, and she reminded me this morning I escaped without giving a seminar, and I guess I'm not going to be able to do that again. So in the way of introduction, yeah, I go by John, and the et al. Is there to remind you. I'm going to be telling you about so many people who have contributed to the studies I'm going to tell you about, and there's going to be many people that are not going to be mentioned. So this is really not about me. It's a story about a long history of a group of people. And in particular, I want to pull out Kendra Turk-Kubo, who was my long-time research associate, and in fact obtained her PhD during her time with me, and then became recently a professor at the University of California, Santa Cruz in my department. And she has contributed substantially to his work. As Fran knows, when he was there, she's really the main driver in my lab and keeps us all focused. So I'm going to start sort of just to put in context the story. This is about marine microbes and their role in biogeochemical cycles in the ocean. And just a reminder that these microbes are really diverse and abundant. They're really important in biogeochemical cycles. We usually think about carbon, but the carbon cycle is linked to the nitrogen cycle. And in particular, because nitrogen is often a limiting nutrient in the systems I study, the open ocean. And so thus the organisms that I'm going to talk about are nitrogen fixers. These are organisms that can take nitrogen out of the abundant gaseous nitrogen in the air and make it available to organisms, are important because they are an external input of nitrogen that maintains the fertility of the oceans as well as the land. And the other point I wanted to make with this slide is just cartoon-like in that we know microorganisms are really abundant. If you look under the microscope, you see a lot of things. The nitrogen fixers, even though I'm going to say they're really important, are actually very low in abundance relative to these other things. So I'm just trying to make the point, if I pick out one of these microbes under a field of view, it might be a nitrogen fixer, and probably you wouldn't even be lucky enough to see one in a field of view under the microscope. So they're very low abundance relative to the main organisms, and yet they're important to the nitrogen input. But it is why this story has been kind of a hard one to approach. So this is what we're talking about, is it's a chemical reaction that's catalyzed by an enzyme called nitrogenase. It's a very difficult reaction. As humans, we do this chemically at great cost. both in terms of fossil fuels and in terms of energy consumption. But it's the reduction of an N2 molecule to ammonium, and it has that chemical reaction done by this very complex enzyme nitrogenase. Does a number of things. It also reduces hydrogen to H2 as a necessary part of that reaction. But the important thing to remember, it's really energetically expensive. It takes a lot of ATP and reductant, and that's why not every organism does it. And the other thing to remember is it's really rapidly inactivated by oxygen. So this is important, especially when you talk about things like cyanobacteria that are fixing nitrogen, because they, of course, evolve oxygen through photosynthesis. So if they're also fixing nitrogen, they have to solve that problem. So it's a very complex enzyme, and the last thing I'll say about it is that it's multi-subunit, actually 2 different proteins with multiple subunits, and one of the important ones is called the iron protein. It's encoded by NIFH, and that's important to you because NIFH was a gene that I targeted in order to go looking for nitrogen-fixing organisms. So nitrogen fixation in the oceans is roughly equivalent to the amount of nitrogen fixation that occurs on land through both natural sources and through agricultural nitrogen fixation. And we now know it's an important, whoops, have to do that at least once. We know that nitrogen fixation is important input to the surface, and it balances the losses due to the anaerobic processes that convert nitrogen back to nitrogen gas, as well as the nitrogen that's lost to the deep sea and to sediments. And so it's an important source. If we didn't have that, we would eventually run out of nitrogen in the ocean. And it supports a large fraction of net community production in the oceans. Okay, so who does nitrogen fixation? Well, microbes do nitrogen fixation. And it's a diverse types of organisms. So archaea, some archaea can do it, some cyanobacteria can do it, some bacteria, non-cyanobacteria bacteria can do it. Whatever they are, they have to have a way of dealing with the oxygen, whether it's the oxygen from the environment, in which case bacteria usually just have high respiratory rates or other mechanisms to protect themselves. Or if they're cyanobacteria, they have a number of mechanisms, either by fixing nitrogen when they're not evolving oxygen, temporal separation, or some cyanobacteria actually have specialized cells, filamentous cyanobacteria, where they keep the nitrogen fixation protected from oxygen. But the point here really is that, for example, when we wrote this book, we said that nitrogen fixation is catalyzed by diverse but limited bacteria and archaea species. Notice that we don't say eukaryotes because this book and all the other textbooks say that no eukaryotes fix nitrogen, only prokaryotes or bacteria or archaea. So the study of nitrogen fixation in the oceans has a long history dating back to Redfield and his analysis of elemental ratios in the oceans, and he really and others sort of assumed that nitrogen was not really a limiting factor in the oceans because if there was a deficit in nitrogen relative to phosphorus, which is only available geologically, it would just be balanced by this nitrogen fixation, even though there weren't many measurements or there wasn't really a good balance of how much or known how much nitrogen fixation was occurring. So for a long time, the oceanographers kind of ignored nitrogen fixation as probably not that important process. And then Ed Carpenter and Doug Capone came along and started studying this organism called Trichodesmium and found high rates associated with this cyanobacterium. That's a side story. It was a controversial point at that time. But it became of big interest suddenly to biological oceanography and geochemists as people realized that actually nitrogen fixation does seem to be a kind of a big deal. And then, as I'll show, a number of people who were trying to balance the nitrogen cycle in the ocean said, hey, but wait a minute, if we calculate up all the nitrogen fixation that we know about and we calculate all the losses, there seems to be an imbalance. There's some input of nitrogen fixation that we don't really know about. And so that spawned this whole thing of people saying, well, where's the missing nitrogen? Where's it coming from? Which is part of the roots of my story, because people were saying, well, maybe there are nitrogen-fixing organisms out there that we just didn't know were there. It's not just about trichodesmium. And so these nitrogen balances claimed that there was an underestimate of the nitrogen fixation process, or the nitrogen input. And that was kind of a motivation for a number of studies of trying to figure out, well, where does this nitrogen fixation come from? So historically, nitrogen fixation in the ocean, as I mentioned, was thought to be largely due to trichodesmium. Ed Carpenter's and Doug Capone's target organism, and many other people, Toshi Saino, as well as these symbionts of diatoms. You can see that these are actually filamentous cyanobacteria. They make that heterocyst that protects nitrogen from the oxygen being evolved from the vegetative cells. And these were basically the things that were thought to be the nitrogen fixers, the big players in the oceans, really up until the 1990s. And that was largely because these were things that you can look under the microscope, and they're big things, and you can throw a plankton net over them, and you can dump the contents into a bucket, and you can actually see the aggregates of them, and they're things that you could actually see. So for a long time, nitrogen fixation studies were biased by the fact that it was focused on these known things that were big enough and easily picked out with a pipette and studied. In the 80s, John Waterbury isolated this thing called Crocosphaera, which is a unicellular cyanobacterium, but that was the first discussion that something like that was an additional player. But it was ignored for a long time. Why? Because it's not easy to see. You basically have to develop a specialized flow cytometer to look for it, because it's not that abundant. So it was kind of ignored for a long time. So that set the stage for going out and looking for other nitrogen-fixing organisms. Because if you're only going to look in the plankton net, maybe you're missing some of the other things. Maybe there are other nitrogen fixers besides trichodesmium and this filamentous cyanobacterium that I just showed you called Richelia. And when I started my career, molecular biology was just becoming a big thing, which probably says something about how old I am. But that meant molecular biology gave you the ability to go look in a water sample for a type of organism without having to see it, or being dependent on expecting it to have fluorescence or not. You just looked for the DNA, for the gene, in this case a nitrogenase gene, and if you characterize that gene sequence, you could tell something about the kind of organism was there. So it's sort of an unbiased way to go answer that question, are there other nitrogen-fixing organisms? And that was done by something called the polymerase chain reaction, which probably these days everybody has heard of because it's used in a lot of biomedical tests, and a way to make that quantitative or count genes called quantitative PCR, and then even more recently, just go out and sequence large chunks of DNA or metagenomics. So those paved the way to amplify and or sequence DNA and quantify abundances of unseen things in samples. So to get to it, 1998, it all began because I started going out and looking for nitrogen-fixing genes. And so this is, you go out with a set of PCR primers, you take a sample, extract DNA, and you amplify up now this specific gene. If you find the gene, you say, yes, there are nitrogen-fixing organisms. And if you sequence it and compare it to the other known nitrogen fixation genes from different types of taxa, you say, well, it's more like a cyanobacterium, or it's not like a cyanobacterium. So the funny part about this, to those of you in the know, actually predates this when I developed the PCR primers, which was closer to the 1990s end of that than the 1998 end of that. There were only 8 nitrogenase gene sequences in the database. I think we passed 30 or 40,000 some years ago now. So it was not that informative, and one of the first things we did is we started amplifying nitrogenase genes from a bunch of known organisms, so we could take the gene that we found in the ocean and compare it to other things and say, Well, it's more like trichodesmium, or it's not like trichodesmium. It's like a bacterium. It's like a cyanobacterium. And what we found was something that we put in this group of sequences, which we called Group A, for lack of anything else to call them, which ultimately became this organism we were looking for called, I call UCYN-A. It's actually an abbreviation of a PCR primer set that Dave Scanlon's group was using for a similar sort of reason. And so this sequence, it's actually literally here, a very short sequence in these days, 350 or so base pairs, told us that there was something that we believed was another unicellular cyanobacterium that we couldn't see under the microscope out there in the ocean. This was done by a technician that I had in my lab, Mark Mellon, who passed away, unfortunately, a few years ago. So we had a new thing to look for, and we also found that not only with this sort of approach, we found that nitrogen-fixing organisms in general were much more diverse than we had previously thought. So this is just a tree, and each one of these branches, sorry the resolution of this is not good, is a different species of a nitrogen-fixing organism. And so these are all the cyanobacteria, these are all the bacteria. This is another unknown story, or a story with an unknown end yet. But this just shows you that there's actually more organisms than trichodesmium and the diatoms and Crocosphaera, one of them being this thing that I'll call it UCYN-A, and that also within each one of these groups, there's a lot of microdiversity of strains. So there's first thing is there's a lot more nitrogen-fixing organisms out there. But one of the challenges in all of this is just the scale of the oceans. So when we try to assess whether there are other nitrogen-fixing organisms and how important they are, we're limited by the fact, and I guess this audience knows, you can only get on so many ships and you can only go so many places and you can only be there at one time. So our sampling of the ocean and being able to assess how important these things are around the ocean is still limited by our ability to do that. So we rely a lot on mathematical models, and this is something provided by Stephanie Dukovics that uses characteristics of known nitrogen-fixing organisms to predict where they are in a more basin-wide or global sense. So the problem with this is that these models depend on knowing what the organisms are and what their characteristics are, how big they are, what their growth rate is, what their phosphorus requirements are, and so on. So that's great for things that are cultivated, but if they're not cultivated, then how do you parameterize the model? So using the molecular biology techniques, a lot of PCR and now the things like the Terra Ocean data sets, we have a better appreciation for the patterns, the biogeography patterns of these different organisms around the globe. This is a summary of a lot of PCR data collected by many different people. There's now a big database paper, second database paper that's come out recently to just kind of put together everybody's data and show where these things are. Well, one of the things that turned out is this thing called UCYN-A started popping up everywhere, even more places than trichodesmium, especially in regions that trichodesmium really hadn't previously been reported. So I'll mention this in a second, but you might see some things out here like in the Arctic Ocean, which was not really thought about as being a nitrogen-fixing area for a long time. And the Terra data sets, again, recently a summary of that using all kinds of imaging techniques as well as metagenomic techniques has helped to fill in this map of where things are found. Now note that imaging is great for, again, trichodesmium and the diatom symbionts because we know what to image, but we don't know what to image for UCYN-A, and that shows up in their metagenomic analyses, which shows that in their samples that there's a lot of UCYN-A, even though, of course, we can't categorize them yet by imaging technologies. So this was an enigma for a long time. We were kind of left with only being able to kind of look for this DNA sequence. There was not a culture of this organism. We couldn't really look at it under the microscope. We thought it was a unicellular cyanobacterium like that Crocosphaera. And all we could do was the experiments we could do. And we could see that the nitrogenase gene was not only in this genome, but that they were actually expressing it, meaning that they were actually making active proteins. So we knew they were alive and kicking. And we knew that it was showing up in a lot of different places, including greater extents in the tropical and subtropical waters, but often pushing into what seemed like they were appearing up in coastal waters as well. So it seemed important, but we didn't really know too much about it, and we still couldn't really figure out what it was. So one of the big changing points came around in 2010, when Brandon Carter and my lab developed a flow cytometry technique to take a seawater sample and sort all of the individual size and fluorescence characteristic cells into different pots. And so this is just showing that you can take a seawater sample and sort specific population into a test tube, and then you can go do an experiment on it. Well, at that point in time, the experiment was, let's determine the entire sequence, not just the nitrogenase gene sequence, but the entire sequence for the organism. If it's a unicellular cyanobacterium, it's going to have photosystem 1, photosystem 2, rubisco, all the biochemical pathways a cyanobacterium would have. And the striking end of the story was that when we sequenced it, it was not at all like what we were expecting. Indeed, the genes that we got looked like a unicellular cyanobacterium, but it was missing most of the genes that you would expect a cyanobacterium to have, even the really important ones, like the photosystem genes that carry out oxygen evolution, for example. And more fundamentally, rubisco, which is carbon fixation, which all cyanobacteria do. And the entire TCA cycle, not just a part of the TCA cycle like some cyanobacteria, but all of the genes in the TCA cycle. It couldn't make its own nucleotides. It couldn't make its own amino acids. And so now we had something like, all of a sudden it was more interesting than I thought it was to begin with. But it left us kind of scratching our heads until we went back and used another flow cytometry technique where we sorted individual cells out of the ocean and looked where this nitrogen fixation gene was and found that the nitrogen fixation gene was always associated with the same type of organism as identified by an 18S ribosomal RNA sequence. In other words, we were able to show that this unicellular cyanobacterium, which isn't really a cyanobacterium, was associated with a specific type of algae, a haptophyte called Braurotosphaera bigelowii. And not only that, but using another technique called nanosims, basically mass spectrometry on a single cell level, we were able to show that those organisms together, as defined by doing FISH, or fluorescent in situ hybridization for identifying the cyanobacterium and the Braurotosphaera, that when you added labeled nitrogen gas, isotopically labeled, or isotopically labeled bicarbonate, that together they were fixing nitrogen, fixing carbon. The Braurotosphaera is fixing the carbon, passing it to the cyanobacterium, or UCYN-A, and UCYN-A was fixing the labeled N2 and passing it very rapidly to the Braurotosphaera. So now the story made some more sense because it was clearly some sort of symbiosis, not unlike that Richelia that I showed you in the pictures of the diatoms that you could collect by net. So it now made sense of how this genome could be lacking so many things because it was a symbiont with another organism. So in summary, kind of here, where we were at that point was we had found this thing called UCYN-A in the open ocean. It expressed nitrogenase during the day, which no self-respecting unicellular cyanobacterium should do because of the oxygen problem. But it doesn't fix carbon dioxide, and it doesn't have photosystem 2 to evolve oxygen. And we learned that from the complete gene sequence, that it not only lacked those things, but it lacked a whole bunch of other metabolic pathways as well. Rubisco, entire TCA cycle, photosystem 2, phycobilid proteins, that's why we couldn't see it under the microscope, etc. So then we found it was a symbiosis, and that sort of explained things. Now, on the other part of the world, on the other side of the Pacific, Kyoko Hagino, who's a paleoceanographer, was studying coccolithophores. And she studied this really interesting one called Broadurosphaera. And she was working on Broadurosphaera, and she was doing these really nice electron microscopy images of the internal structure. And this was the same time. And so our paper came out, and she was just then writing up this paper, and she was able to take our information and go look at this body and find out that, in fact, that thing that she was calling a spheroid body was actually what we were calling UCYN-A. And so it all made sense. This spheroid body was really UCYN-A, and she knew a lot about the biology of this because she's a paleoceanography, and she knows about this, and she knows that this comes in all kinds of different forms, and also that they have complex life cycles. So she was studying this because she's a paleoceanographer, and she wanted to look for it in sediments initially, but she was able to realize that it probably had a different stage, and she ultimately was able, by sampling at a different time of year, to isolate in culture the what's called the naked form that is covered by organic scales instead of calcified plates. And she obtained a complete xenic, not axenic culture of this. So this will be part of our story moving forward. So after at this juncture then, that's about the time that a lot was happening in Terra, and some people sitting in this room I got an email from Sylvia Cenas, who I hadn't seen for many years, telling me, hey, guess what? And it turns out that a couple of bright students working with her and Ramon Massana had found sequences in the Terra metagenomics and also had developed fluorescent in situ hybridization probes to be able to look at the Broadurosphaera and the UCYN-A. So this was a remarkable thing, and it was really the first time that we were actually able to see it. I've been telling you it was difficult to see these other organisms other than Trichodesmium. This was the first time that we actually were able to see it. And what we saw collectively is that this thing is not just one thing, it's different things, and they come in different sizes. And therefore, the genes would be collected in different size fractions. So this showed now the sort of the complexity that we're really talking about different species. The species, as it'll turn out, that Kyoko cultivated was one of the larger forms. The thing that I probably found in that sequence back in 1998 is probably one of the smallest forms. But they're closely related. And they make little clusters or families if you look at their gene sequences. And you can predict which Broadurosphaera type is associated with which UCYN-A by their gene sequences. So there's a different 18S signature for Broadurosphaera with the UCYN-A1 as compared to the one for UCYN-A2. And what we've been able to show, and Kendrick Kubo has a couple of papers on this, is that one of these is really a more oligotrophic open ocean ecotype, and the other is more of a coastal form. And they've co-evolved with their Broadurosphaera hosts. So the story just continues to get more complicated, and meanwhile, we start seeing that this thing is just really important in many, many, many different places, including coastal waters, which was never really thought to be an important place for nitrogen fixation to occur, and even in the Arctic Ocean. Shiozaki in Japan saw that there were sequences there, but my student Katie Harding also had a PNAS paper where she showed that not only were these UCYN-A sequences there, but by the nanosims technique to show that they were really actively fixing nitrogen. So they weren't just carried up there as dead cells, they're actually fixing nitrogen. So whatever this thing is, I'm going to tell you, but now focus on the biology of this thing, but they're somehow globally important in nitrogen fixation worldwide. So where are we? We knew this thing was something that only fixed nitrogen during the day, but now the enigma is it's doing that, but it's inside a cell, which is evolving oxygen. So it still has to deal with the oxygen problem. And we know that it has this really unusual metabolism that's lacking many synthetic pathways. So if you do an analysis and you can take what the algae, the Braurosphaera genome can provide, and you do an analysis of what the UCYN-A provides, you can actually map using something called flux balance analysis what things have to be made by the host to provide to the cyanobacterium in order for it to be able to survive and grow and divide. And what you find is that it's many, many, many specific compounds that have to be provided by the host to fix that broken metabolism. Somewhere around 30 different molecules have to be provided to this thing. That's a lot of dependencies for this thing that we were calling a symbiont. So that sort of brought up the question of, well, maybe it's a little bit more than a symbiont. Maybe it's actually an organelle, more organelle-like than a symbiont. And in history, in biology, we really know of only 3 cases where an endosymbiont has led to an event of evolution of an organelle. The chloroplasts and the mitochondria, of course, where we have consumed a bacterium or a cyanobacterium, and they reduced their genomes and they became a symbiont. And then this interesting protist, Polynella, which is a heterotrophic organism, which more recently has taken up, again, a cyanobacterium, analogous to what happened with the chloroplast. And they have co-evolved such that this cyanobacterium has gone beyond just being an endosymbiont. It's lost a lot of its genome. It actually imports proteins from the host cell, from the protist cell, to fill out its metabolism. It's actually exchanged genes. Some of the genes from the cyanobacterium got integrated into the host genome, and so on. So maybe UCYN-A is more like that. We started acting ourselves. And people talked about the possibility of this for a long time. And there's a paper with John Raven a long time ago that they actually called those heterocyst or filamentous cyanobacterium nitroplasts because they're acting like a nitroplast, even though it's not really been shown that they're an organelle or have the features of an organelle. And people were even making arguments for why there isn't a nitrogen-fixing organelle due to the geological history or geochemical history. So the idea is out there that there should have been, or there could be, a nitrogen-fixing organelle. Okay, so Fran just told you he came and visited me very shortly after he got there. He came to my lab very excited and said, I've been looking at the samples that we have for doing this fluorescent in situ hybridization, and I've been actually measuring the Braurosphaera cell and measuring the cyanobacterial cell. And, it's really interesting, but as one gets bigger, the other gets bigger. And so we've got examples of 3 or 4 different sizes, and they all seem to be kind of correlated somehow. And Mick Follows, I was collaborating with for other reasons. We sat down in the office. I think Fran still has the picture on the board. We were talking about that, and Mick is, well, there may be a reason for that. So this went on to a very interesting study, and it just shows amazingly how linear this data is, the UCYN-A size to the host size. I mean, this just doesn't happen experimentally very often, not in my lab. And the reason for that is that actually happens with organelles. So if you look at chloroplast biovolume with size of cells, and this is haptophytes, but it's actually true in general of phytoplankton, they scale, because there's a metabolic dependencies in between the two that result in that scaling. So Mick and Kei Inamura developed a mathematical model for the interactions between these two cells on the basis of carbon supply, nitrogen supply, and the oxygen dynamics involved with photosynthesis. And we're able to show that if you modeled this metabolism as a function of the ratio of the sizes, that the growth rate of each crossed at a maximum growth rate of about predicting a ratio of the sizes of the two, basically what Fran was measuring. So that's an interesting observation, but the most interesting part of the observation is that's basically what happens with organelles. And so it was another argument that, hey, there's something a little more intimate here in this relationship than a simple endosymbiosis. So how do you define endosymbiont? What line do you have to cross to? Well, it's a little bit of an argument. It has a long history of what makes something an organelle versus an endosymbiont. But some of the things that have been used, for example, in the case of that Polynella protist in the chromatophore, is that either there's gene transfer from the endosymbiont to the host genome, that there's tight integration of the cell division, because if there's not tight integration, then you lose whichever you want to call it, the endosymbiont or the organelle. So there has to be coordination, just as there is in division of the chloroplasts and the mitochondria. And some sort of metabolic integration, a dependency of the metabolisms between the two, which can be that the host actually makes proteins that the organelle can't, and that the organelle just imports those proteins from proteins that were encoded by the cell's nucleus, not its own genome. Well, we had previously some evidence that, in fact, they did coordinate their daily growth and division from doing whole genome expression arrays. This was done by Marin Munoz Marin, another Spanish postdoc that predated Fran in my lab, also a Marie Curie fellow. But she saw that the expression of division genes in UCYN-A had this cycle that looked like it was coordinated with the division of the host, and could actually see evidence of some division of the cyanobacterium. But that was kind of all we knew at that time. So, does UCYN-A have the features of an organelle? How does it divide in relation to the host? And has there been gene exchange or protein import from Polynella? Well, we haven't been able to get the entire genome of the host yet to be able to answer this question. But we were working on this question, as I will show you in a minute. I was introduced to Carolyn Larrabell at the Advanced Light Source at Lawrence Berkeley Laboratory, and she works with soft X-ray tomography. She actually developed this microscopy technique to be able to look at cells and their internal structures using the Advanced Light Source. And so we collected samples from this culture that Kyoko Hagino had provided, and we're able to reconstruct what a cell looks like. So this is a movie of really what the data looks like. It's three-dimensional data because they scan through the entire cell, looking at absorption coefficients, and the absorption coefficients basically measure the density of molecules so that they can identify the different organelles. And so what this ended up showing is that we have a really nice organization of the cell. This is the nucleus. This is the UCYN-A. Mitochondria are wrapped around the UCYN-A, which makes sense for various reasons that I won't get into. And so for the first time, we had a really nice image of the UCYN-A cell. But even more intriguingly is we were able to put together over a diel cycle how this structure changed. And so this is the same thing now, looking at a cell that's actually dividing. And so you can see now in this image that UCYN-A is divided, the chloroplasts are dividing, the nucleus is starting to elongate, as if they're a carefully organized dance of separation. And in fact, that's what we could show, is that there's this coordinated sequence of division through the life cycle of the cell and its, well, I'll just say it, its nitrogen-fixing organelle. So it starts out with the UCYN-A or dividing followed by the chloroplast dividing, elongation of the nucleus. The two cells begin to separate, and the UCYN-A is kind of the last part of the cell to separate. So very, very carefully synchronized, just as one of the other organelles in the cell. And then along came, we were just finishing that study when Tyler Cole came to my lab from Andy Allen's group. And we had been struggling with trying to answer this organelle question for a long time. And there was really two questions, either the genes in the genome of the host, or are there proteins from the host getting into UCYN-A? So he did some really, again, using flow cytometry, some really elegant development of a technique so that he could break open this cell and separate a pure preparation of the UCYN-A body or organelle. So this is the intact symbiosis. He breaks it apart and then purifies UCYN-A, not unlike the experiments we did initially to find what we thought then was a symbiosis. And then you can send this preparation out for proteins and determine all the proteins in it and identify what kind of proteins they are and whether they come from UCYN-A's encoded by UCYN-A's genome or whether they're encoded by Broad-Rootosphaera genome. And you can take this apart and you can again do the proteins and you can find out which proteins are UCYN-A encoded and which ones are Broad-Rootosphaera encoded. And the surprising thing, which was jaw-dropping for me in my lab meeting where he presented this data, is that about half of the proteins that he found in the UCYN-A were encoded by Broad-Rootosphaera. And the only way that can happen is if the proteins are encoded by Broad-Rootosphaera or encoded in the nucleus of Broad-Rootosphaera. They're transcribed, they're translated in the cytoplasm of Broad-Rootosphaera, and then somehow they get into the UCYN-A. I'm not going to call it a cell, I guess it's an organelle-like. Well, in Polynella, the way that works is the same way as in mitochondria or chloroplasts, is that there are specific targeting sequences that go on the end of the protein that identify it to go to that particular organelle. And so the capstone of the story was that when he looked at those proteins that had been encoded by Broad-Rootosphaera, looked at their sequences, most of them had a specific previously unidentified motif, the size of a tag or targeting sequence, leader sequence, to be imported into UCYN-A. So if you look at all those proteins, about half the proteins not encoded by the UCYN-A genome, they have a specific leader sequence that intracellularly are targeting to the UCYN-A, just like an organelle. So it's a distinct sequence from that kind of sequence that sends proteins from the cell to the mitochondria or the chloroplast. It's a unique identifier, like a postage stamp, I've heard Tyler say, to send them to UCYN-A. So this is really now conclusive evidence that this is something beyond endosymbiosis. It's actually something that is at least an early stage organelle, nitrogen-fixing organelle, or a nitroplast. And this was just published a few weeks ago in Science, and it's been all over the news ever since then. It made a big splash. It's been very, very popular, still coming out in various news outlets, as well as all over Twitter and Instagram, and there's even a YouTube or two about it. So it's been very exciting. Some of them are very amusing reading, but it's a very exciting time, particularly because people are saying, well, one of the things that has been a focus of nitrogen fixation research for many decades is, well, if we could just get agricultural plants to fix nitrogen, well, then we wouldn't have to fertilize at this costly, environmentally costly, fossil fuel costly. And they've still been working on it, but it's like, well, maybe it's just not going to be possible. Well, this says a photosynthetic organism can have a nitrogen-fixing organelle. So there's been a lot of interest, not only from the basic biology world and evolution world, but the people who are now trying to figure out how we might use this information or organelles like this to make plants. I think that was probably one of the first emails I got was from a group like doing that kind of work. So lots of questions, plenty to do. This is the first known nitrogen-fixing eukaryote. I have to call it a nitrogen-fixing eukaryote now, because if it's an organelle, then that means that the thing that's fixing nitrogen is actually a eukaryotic cell, not a cyanobacterial symbiont. But, I told you about that culture, which we could do this experiment, but it's not necessarily the case that all the other strains would have to be at exactly the same stage of evolution. And we need to figure out this mechanism. So are there membrane transporters that identify that targeting sequence that take up those specific proteins? And ecologically, we still can't answer the question, how important is B. bigelowii compared to trichodesmium or the diatom symbionts? Is it twice as much nitrogen fixation? Is it half as much? Because of the space and time scales, it's a really hard thing to answer. Are there other nitroplasts? Maybe this is just the one. Remember, I discovered this because I started with a gene sequence and have doggedly for decades tried to figure out what the thing was. So if I had been starting top down, it might have been a while before we sort of realized what we were at. And the only evidence or the answer for that I can tell you is that, again, for decades, there have been in freshwater systems, there's these spheroid bodies that occur in diatoms now, not haptophytes. And I've known a lot about them, but they've not received as much attention as this. And so and now there's some people working on freshwater. This was just published possibly within the last week. Recently, a marine isolate of a similar thing was isolated from tropical waters. And these things could be potential organelles as well. But further study is necessary to really figure out how far along the path of evolution they are. So there will be more surprises to come, and I couldn't really cover all of them. But with that, I'd like to thank a number of people and funding agencies. There's again, Tyler and Kendra, Esther Mack, who's also an author of paper, Britt Henke, who's my technician. I'm very appreciative of all the work that they've put in, as well as the funding agencies, the Simons Foundation and the Gordon Betty Moore Foundation. I'd just like to call out David Kingsbury, who funded me back in the early 2000s and actually made it possible for me to have access to the photocytometer that made this original discovery. And Marion Carlson of the Simons Foundation, who invested in us most recently and actually enabled us to work with Kyoko to get that culture in place that we could do these kinds of experiments. And with that, thank you very much. And I'm just pointing out that I'm going to have to go rewrite my book, but a lot of other people are going to have to too. So thank you very much.
Thank you very much for the talk. Is there any question? Okay.
Thank you for your talk and your time. The ecological success in coastal waters, how would you explain for the UCYN-A in symbiont this? What are your hypotheses and so on?
About why it fixes.
Why it's so abundant? Yeah.
I don't have a good one. I mean, one of the things that I didn't have time to talk about is that Kenda Turk-Kubo and Matt Mills discovered that the host seems not to be able to take up nitrate. So using the same technique, nanosims, it won't obtain label from isotopically labeled nitrate. So somehow, which might suggest that that host is evolving for specific niches as well, which might mean that it continues fixing nitrogen when they're in the presence of fixed nitrogen. And fixed nitrogen in coastal waters would be one of the reasons you would expect it not to fixing. So that's the only thing I can suggest as maybe being part of the explanation.
Some other questions? Yeah.
Very nice talk, congratulations. I am wondering, the coccolithophorids have sexual reproduction also. So I don't know what happens with the nitroplasts when these organisms goes to diploid and haploid cycles.
Yeah, that is actually a great question. It's also, so Kyoko, one of the things that Kyoko Hagina has been working on has been trying to catch the thing mating and see what happens during the mating cycle. And I have to say, it's just really frustrating to do these experiments, and they die very easily. And she's had a few observations where she's seen things started to get together, and we've actually seen some things on the imaging as well, but we've not been able to complete the life cycle, so we don't really know. And in fact, we don't even know which of these two forms is haploid or diploid. I mean, we could make a guess, but we don't really know. So
to know if this haploid or diploid. Yeah.
yeah, that would be great. Yeah.
Other questions?
Hi, John. Oh, it was impressive. Thank you for your time. I have actually two questions. One is, why an aptophyte? And now you're showing examples. Probably some diatoms have a similar thing still to be demonstrated. But is there any specific? I mean, is there anything that make aptophytes especially suitable or suited for this to evolve in terms of timing and evolution, in terms of physiology?
I don't think there's anything like that. I mean, the thing we've tossed around is, number of haptophytes are predatory, and this whole event might have started with ingesting. In fact, the body ends up being next to a feeding vacuole. And in fact, my other student, which I wasn't able to present, but Esther Mack has actually shown that not only is this thing doing this, but it's also eating bacteria in the media, so it's also feeding. So it might be something about ingesting. A lot of phytoplankton ingest, though, so that doesn't really make haptophytes that special. But it's maybe something of that. And again, maybe the answer is really we haven't looked enough. Because again, I don't know we would have seen this if we hadn't started with the gene, because there's not much to see. There's an unidentified blob. And in fact, the one, we still haven't actually seen the one that from the original discovery, because it's a factor of 5 smaller. And so trying to see this body in that, probably, I don't know when or how that, probably some advanced imaging technology, somebody might get it, but otherwise it'd be really hard to see. So maybe we're missing other events. Maybe it's not so special.
And my other question is about the non-cyanobacterial diazotrophs. You left them aside just because it was not the main flow of your talk, but can you say something about them? Is it still, we're still struggling to see whether they make an important contribution to, or any contribution at all to nitrogen fixation, right?
Yeah, so that's a long story. So the same time we found this sequence, I found bacterial sequences reported in the same 1998 paper. And I just ignored it because it was too hard to study. In more recent times, a lot of people have picked up on that, and of course, we're finding that there's tons of nitrogenase sequences around. And one interpretation of there's a lot of nitrogenase sequences is that there's a lot of important nitrogen fixers. But another interpretation is there's a lot of genes that are carried around that are not necessarily used. In my early studies, we went to a lot of habitats like the hypersaline lakes, Mono Lake, and Chesapeake Bay, which is not really a nitrogen-fixing environment. And we just found incredible diversity of nitrogen-fixing, these non-cyanobacterial nitrogen-fixing genes. So I just kind of personally had decided that they're not likely to be important. Now, there's huge databases of these genes now, and some of them we know are actually transcribed. So they must be active. Some of them must be active. We tried to do an unbiased technique with nanosims where we just looked through a whole filter looking for hotspots that could be non-cyanobacterial diazotrophs. We didn't get that many. It's not a perfect experiment. So at the end of the day, it's unresolved. You know, there's not a lot of evidence that they make up a huge fraction of nitrogen fixation, but there's certainly evidence that some of them are probably fixing, particularly in particles or environments where you could get around the oxygen problem and have enough organic matter food. But, you know, in my mind, it's still an unresolved problem. And that's one of the problems that Kendra Turk-Kubo has been focusing on now.
Well, thank you very much for your talk. I think that just following up a little bit on this, most ocean models that have some biogeochemical part in it use nitrogen as the limiting element, right? But some early oceanographers were already saying that phosphorus should be the limiting element because you have such a huge source of nitrogen, atmospheric nitrogen, and there would be some way that this is going to be used somehow. Yeah. And so now that you see this widespread potential for nitrogen fixation, you just mentioned that maybe it's not so large in terms of nitrogen fixation. The potential is there, but does this put phosphorus back in the spot? That's the limiting element for the ocean, because there's also nitrogen fixation in coastal areas and everywhere now. Yeah. So,
you know, I'm not sure if it changes the bottom line or not. You know, the facts are that argue for nitrogen limitation is almost anywhere you go in the open ocean, you do an experiment. In the oligotrophic open oceans, you add nitrogen and you get a response. So that argues that at least at that time and place, in that sense, nitrogen limits productivity. But I think in the larger sense, I'm not sure that the discovery of these different organisms changes things much, because all of those biogeochemical arguments about the missing nitrogen and all that, they were all done by a biogeochemical measurement of the N to P ratios, fixed N to P ratios in the oceans, and trying to make estimates for how much, and those estimates don't care whether it's 1 organism or 10 organisms. They're measuring nitrogen fixation as an unbiased ratio of elements generated by nitrate and phosphate. So, and I'd say that, you know, the whole argument about the balance is, well, the errors in the estimates were greater than a lot of the differences between the nitrogen inputs and outputs. So I'm not really sure that there really is that much of a imbalance, certainly not over long time scales. And I don't think that really addresses the answer to your question, but I don't think these non-cyanobacterial diazotrophs or the other diazotrophs will necessarily change the answer to whether it's nitrogen limited or not. And just because they are active, that doesn't mean that they are actually fixing enough to adjust the ratio, right? They're just there, they're fixing nitrogen, they're doing it because it's ecologically possible for them to make a living doing that, but that doesn't necessarily mean that they're going to top up the imbalance or not top up the imbalance.
But it's costly energetically, so I mean, there has to be a good reason.
Yeah, But that doesn't mean that doesn't, I guess, so I'm arguing the other way around, that just because these things are fixing nitrogen doesn't mean that they are going to fix the ecosystem ratio of N to P. They're going to get by, and maybe for the reason you just said, maybe they can't necessarily adjust the ratio. I mean, at the end of the day, I mean, it has to be true in some sense, right? I mean, there's a geological source of phosphorus, but there's an atmospheric source of nitrogen, so there's always going to be a possibility of nitrogen inputs. It might be a matter of how long it takes for them to be able to top it up, but maybe that's why over long time scales it does.
Thank you, I appreciate it.
Okay.
Thank you for your talk. It was very interesting. I wonder if you could try to trace back when this event, the first time it happened, like we managed to do this for mitochondria and chloroplasts, so maybe when the first endosymbiosis with the nitroplast happened in time.
Yeah, I'm going to let Fran answer that because he thinks about this a lot. I mean, I could give you my summary of his answer, but he has a different resolution than me. I'd say 100 million years ago. He's going to tell you, they figured out that it's not 80, it's 120. So, but it's sort of in that scale, based on the phylogeny of the things that we have sequences for.
And does it make sense? Was there like a depletion in nitrogen around that time? Yeah, okay.
Because he's thought about this a lot more than I have, and I know he has a ready answer for this.
Yeah, it was basically the lowest nutrient concentration in the ocean around that time, so that kind of fits how these and other nitrogen-fixed symbiosis started about at the same time. So probably it's related with the low concentration in the ocean. Yeah. So, okay. I'm increasing my daily steps now.
Thank you so much, John, for coming first, and second for this beautiful history of the UCYN-A. That to me is going to be difficult now to call it nitroplast because it's been a long time calling this way.
Yeah,
and
all of us.
Yeah,
so
that's
problem.
the curiosity. The first, when for you was the right moment to say, okay, this is an organelle? So because I know you that you need to have all the pieces just perfectly fit in order to make this, open paper.
It was really this protein argument. Because, in Fran's work, we kept saying, it sure looks like it's behaving as a nitroplast. The reviewers would hardly even let us say that. I mean, no matter what we did, we didn't back up enough for the reviewers. We just said it's behaving as a nitroplast. And then, the synchrony of division. Well, it's a beautiful thing, but, that's not really the capstone. But how do you get proteins from the cell into the cell and then to have actually evolved this leader sequence to target it? And those of you who've worked with me know that I'm really skeptical. And when people in my lab present data, I'm like, yeah, but what about, what about? And when Tyler showed this data, I couldn't come up with anything. There wasn't any way it could be contamination. There just wasn't any way to explain it except that. And that was when I said, well, I guess, to be honest, I'm still kind of waiting for a backlash of somebody writing the paper that says, you stupid, But that was really the piece, the capstone piece for me.
I think now is a great moment to look for new nitroplast. And I think all people will jump into the field. So that is amazing because you make that marine symbiosis and the UCYN-A become like a major biological discovery.
Yeah.
So
Thank you.
Wonderful.
It's very difficult to write papers now because we have all this terminology we use, and editing the paper is very complicated now. So.
I think this is going to be the last, before the last.
Well, thanks for the amazing talk. And, well, a bit following up on the discussion about the nitrogen limitation, I wanted to ask you, what do you think would be a proper definition of nitrogen limitation, right? Because we tend to think of limitation as a matter of concentration, stock, but it's perhaps more a matter of the supply rates, right? And also of the organisms that are using the nitrogen or fixing the nitrogen. So now we say if we add nitrogen to the system, there is a reaction. Yes, but by some organisms only, perhaps not by others. So how do we define this properly to be then able to implement this, for example, in model parameterizations?
You know, everything's complicated, I guess. But I think at the end of the day, though, unless you can show that the presence of nitrogen fixers can result in a biomass or productivity response equal to that of adding fixed nitrogen, then you're still, I think you'd still have the same interpretation that the system production or productivity is going to be limited by nitrogen. Because I don't think, for the energetic reasons, that if there's nitrogen limitation, that nitrogen fixers are actually going to be able to compete well enough that they are going to be able to assume the rate of production that was occurring with the non-nitrogen fixing organisms. And in fact, that's what the Darwin model, the McFarlane's models kind of show, is that if you don't parameterize the nitrogen fixers as having deficits in other abilities, like growth rate, or that they just take over the world, and they don't. So maybe things haven't changed that much. I mean, certainly the way you think about it has to change, but maybe the definition that doesn't change. But yeah. Yeah.
I think it's time to go for lunch, but before, because I was the one inviting him, I have the right for at least 2 questions. So the first one is concerning the leading sequences that UCYN-A is using for importing the proteins from the host. Do you think, or did you analyze whether the chloroplast and the mitochondria are kind of, I mean, in this context of Brachysphaera, if they are using the same sequences?
No, they're not. They're unique sequences. So Tyler should be here to answer this, but we haven't. I don't believe there's any proteins in UCYN-A that have chloroplast or mitochondrial leading sequences, number one. And this sequence is unique, so it's not likely that they're imported into chloroplast. Okay. Or reverse. I think that's...
Yeah. That was the question. And the second one is, how do you feel now that you just realized that you are a protistologist?
Yeah. Yeah, well, it's been a problem all my career, is that, you have to, all of a sudden I'm looking at eukaryotic algae instead of cyanobacteria. That was the first problem. Yeah.
Okay, I think that is enough for today, so thank you for this talk.